Welcome back to the foundations of general emergence mechanics. In our last explainer, we looked at chemical bonds, framing them not just as lines between atoms, but as local curvature distribution solutions, where coherence actually becomes form. But today we're taking a massive leap in scale and complexity. We're moving away from those local bonds and diving headfirst into the giant dynamic world of macromolecules. We're talking proteins, DNA, RNA, structural polymers, and membranes. Our main focus for this session? Figuring out exactly how these massive structures manage to fold, function, and hold themselves together without just losing their identity to internal friction.
Now, a macromolecule isn't just a regular molecule that's been supersized. It's playing an entirely new survival game. Think about it. A small molecule really only has to ask one simple question. Can this local structure hold? But a macromolecule, it's constantly asking, can this whole moving landscape hold together while remaining useful? Because it's packed with thousands of degrees of freedom. It twists, it loops, it hides surfaces, it exposes charges. So the challenge isn't just about holding a static shape anymore. It's about maintaining an entire coherence system.
Here's our roadmap for today. We'll go from local bonds to landscapes, see why folding is really friction minimization, explore the dynamic coherence volume, look at misfolding in the active environment, break down biological strategies for persistence, and finally, look at moving without losing itself.
All right, let's kick things off with Section 1, from local bonds to landscapes. A huge trap we often fall into is picturing macromolecules like proteins as these frozen, static sculptures. But in reality, they're anything but. These structures bend, they twist, they loop, they expose different surfaces, meaning their function absolutely depends on controlled motion. Just think about it. A protein only works because it's stable enough to hold an active site, right? But it also has to be flexible enough to actually guide a chemical reaction. A receptor is useless unless it's stable enough to recognize a signal, while still being flexible enough to change its state to pass that signal along. Even a motor protein has to preserve its identity, but stay flexible enough to literally step forward. The takeaway here, biological function demands movement.
Moving right along to Section 2, folding is friction minimization.
Picture a really long chain of amino or nucleic acids. It has millions, sometimes billions of possible configurations. But here is the real secret of GEM. The fold that actually survives isn't simply the one that's physically possible, it's the one that's admissible. The surviving fold is the one that minimizes friction and manages to remain coherent at an acceptable energetic cost. So folding is not simply a shape problem, it is absolutely a friction minimization problem, first and foremost.
To persist, a molecule really has to balance a whole host of elements. We can look at this through a specific GEM schematic that calculates the total cost of maintaining a shape, which we call F total. Now, we're not using this to replace detailed molecular dynamics. Instead, this gives us a much deeper language for understanding persistence itself.
Let's actually break down that calculation. You've got the cost of stretching or compressing bonds, that's F bond. Then the cost of distorting angles, F angle. F torsion is the energy spent twisting around rotatable bonds. F contact is the cost, or maybe the benefit, of different local regions of the molecule bumping into and interacting with each other. F solvent is the penalty for exposing or hiding certain parts to the surrounding medium. And finally, you have F friction, the actual literal cost of the molecule moving through its own crowded internal landscape. For a fold to persist, the sum of all these costs, the F total, must remain below a critical limit, or F crit.
Which brings us to Section 3, the dynamic coherence volume.
Remember, we are not dealing with rigid objects here. A functional macromolecule is a dynamic coherence volume. Picture a folded chain settling into a deep valley in a landscape. It's not frozen dead at the absolute bottom of that valley. Rather, it's stable enough to hold an active site, but flexible enough to guide a reaction. It breathes, it shifts, it opens, and it closes around a stable basin. Honestly, if it were perfectly rigid, it would be dead.
But there's a really delicate balance to strike here. Too much stiffness kills function, but too little destroys the molecule's memory of its shape. So we use the GEM triad to understand this perfect balance for controlled, flexible structure. Think of depth as the steepness of that valley we just talked about. It gives the fold tolerance to absorb energy without breaking. Stiffness gives the fold its specific identity, keeping it from just dissolving into random noise, and viscosity dictates the fold's recovery, how smoothly it returns to its functional shape after it moves. If a molecule has too much internal viscosity, it gets bogged down and trapped in the wrong form. And that leads us perfectly into our next point.
Section 4, Misfolding and the active environment. In GEM, misfolding is a really dangerous friction trap. You see, a misfolded molecule hasn't necessarily lost all of its structure. It's simply found the wrong structure. It's stuck in the wrong valley, and escaping that valley is incredibly hard. This is a vital lesson for us. A locally stable state can actually become globally harmful. A misfolded protein might expose sticky surfaces, aggregate with other molecules, seed even more misfolding, and violently resist being cleared out. Basically, misfolding is persistence in the wrong basin. Useful persistence, on the other hand, has to support the larger coherent system.
So, why do they fall into these traps in the first place? Well, we have to remember that the surrounding environment is literally part of the admissibility landscape itself. It's a foundational rule. No macromolecule folds alone. Water, for instance, is an incredibly active participant in this whole coherence economy. It is definitely not just a passive background liquid. Water acts as the active landscape that dictates which folds can actually survive. It shields electrical charges, it enables vital hydrogen bonds, and critically, it severely punishes exposed hydrophobic or water-fearing surfaces, basically forcing the molecule to fold those parts inward. The surrounding water, the ions, the pH, they literally redraw the map of what is admissible.
Let's look at Section 5, Biological Strategies for Persistence.
When you look across the board, different macromolecules execute uniquely specialized strategies for dynamic persistence. Take DNA, for example. It represents persistence with controlled access. It has to preserve the genetic code perfectly, but still remain flexible enough to be unzipped, read, copied, and packed away. RNA, on the other hand, is persistence with flexible function. It can store information, sure, but it also folds into shapes, catalyzes reactions, and binds partners. Then you have proteins. They are persistence with shaped action. They're the folded coherence tools that do the heavy lifting in biology. And finally, membranes represent persistence with bounded exchange, making controlled physical separation possible. That's 4 different macromolecules utilizing 4 brilliant friction-minimizing strategies.
And that brings us to our final section, Section 6, Moving Without Losing Itself.
As we've seen, a truly useful macromolecule doesn't just occupy a single fixed point in space. It occupies a functional basin of nearby admissible forms. And this is balanced by what we call conformational admissibility, or ACONF.
Looking at this expression, Q represents the actual conformational coordinates, essentially the physical shape of the molecule. U of Q is the raw structural cost of physically holding that specific shape. And gamma? That is the frictional cost of actually moving through that conformational space. For a functional fold to exist, this combined cost has to stay below a critical limit, acrit, across a useful range of motion. The molecule absolutely has to be able to move within its basin to perform its function without escaping into an aggregation trap or completely collapsing on itself.
Zooming out to the grander narrative of life, we can see the steps perfectly building on each other. We started in step 1 with bonds, which allow for local curvature sharing. Now, here in step 2, macromolecules represent the very first organic web, controlled, flexible structure. They teach us the vital lesson that structure must move without losing itself. And this sets the stage perfectly for our next explainer on step 3, the cell, the incredible moment where life actually begins to draw physical boundaries and shield gradients.
I want to leave you with this final, somewhat provocative thought. When we look at the sheer, mind-boggling complexity of a folded protein, we really have to ask ourselves, is biological form built by design, or is it simply filtered through the tiny fraction of folds that can actually remain coherent against the crushing forces of internal friction? It's a profound shift in how we view the physical machinery of life. Thanks so much for watching this explainer, and I'll catch you in the next one.